Welcome everyone to the virtual SMA conference SMA Clinical Trials webinar. This is Jessica from Cure SMA's Family Support Department, and we thank you all for joining and sharing part of your day with us. We would like to thank our national presenting sponsors for their generous support of the 2020 virtual SMA conference, Vexus, Biogen, and Genentech, and we recognize the support of our visionary sponsor, Ionis, platinum sponsor, Scholar Rock, and silver sponsors, Acredo and Permobile. We appreciate all of the many questions we received in advance of this webinar, and we will try to answer many of these during the presentation. You're also able to submit questions throughout the webinar using the chat box feature, which you'll find located on the right-hand side of your screen. Please note that all lines will remain muted during the webinar other than for speakers. And now we'd like to welcome our speakers, Dr. Castro, a neurologist with University of Texas Southwestern, and Dr. Brandsema, child neurologist with Children's Hospital of Philadelphia, and they will be presenting on SMA clinical trials.
Good morning, or good afternoon, or good evening, depending on where you're joining us from. I'm John Brandsema, and I'd like to start by thanking Cure SMA for the opportunity to speak on this topic today. I know it's something that my patients often have questions about when they come to the clinic. What my role is today is to just set the stage for what participating in research is like in general, and also how this applies to SMA. And then Dr. Castro will be going through some of the opportunities right this moment that might be available for participating in research, as well as what is on the near horizon. If we can go to the next slide, please. So these are my disclosures. You'll have them in the conference booklet as well. To the next slide, please. When we think about trying to design a clinical trial, there's a lot of pressure on industry to produce results. If they're not seeing the effect that they're hoping for, a program will be terminated very easily because they're trying their best to bring things into the clinic to benefit as many people as possible. And so some things that can help this in diseases like SMA are a few modifications to the standard trial design that we tend to have. Over 30 years ago, there was a designation made called Fast Track. And this is something that's meant to expedite research for diseases that have a critical need, that have very few therapeutic or no therapeutic options, and have a severe morbidity or level of symptom burden. And so many of the therapeutic approaches in the SMA space have gotten this Fast Track designation. Just under 20 years ago, there was an additional modification to this called Orphan Drug Designation. And this helps in the context of the Rare Disease Act for diseases like SMA, make sure that clinical trials are more feasible. We don't have the numbers to be able to hold trials that have thousands of people in them the way we can do in heart failure or in diabetes research, for example. And so the orphan drug designation allows for a modified approach in terms of numbers and the way that you design a trial and the level of evidence that's required for the FDA to review a medication. But the basic point is to try to design a trial that has the ability to show an effect of what you're trying to do an intervention with. This is much more complicated than it sounds at first glance. A disease like SMA challenges us because there is a wide variability in how people experience the symptoms. So you may be able to have a trial in infants living with early onset SMA where you might be able to see a difference very quickly. But if you go to a later stage of the disease or a milder form, like people who are walking with SMA, for example, there's longer periods of time where they would change naturally with the disease. So unless the intervention that you're making has a big difference in terms of the effect, you may not actually see a difference for a long period of time. And that leads to a very impractical trial design where you have to follow for years and it's very expensive. And so sometimes we choose to focus on specific populations for that reason. If we can then go to the next slide, please. So one of the key pieces to trying to ensure success is the inclusion-exclusion criteria for a trial. And this is something that can lead to a lot of angst for the providers involved and also for the patients and their families, because it's really heartbreaking when you're just 1 month short or a little bit too old or have a little bit too strong of a breathing test, or there's something about these screening criteria that makes you not able to participate in a research trial. But these are chosen very carefully because if we don't ensure that the population that we're enrolling in a trial is one that we're likely to see a difference in, it's really to the disadvantage of everybody to do it that way, because you're not going to see an effect the way that you're hoping to, and the medication won't come to people who need it as quickly. If you're interested in knowing what the inclusion and exclusion criteria are for a trial, this is one of the chief things you need to know if you're interested in participating in research. And there's many resources that are available to you. Cure SMA has a description of ongoing research on their website, and people within the organizations that are very happy to connect with any families who have questions about this. The Muscular Dystrophy Association, or MDA, also has a clinical trial finder tool that you can access related to SMA research as well. But I think one of your best resources for this is talking to your provider, and most likely your neurology provider, although there are other specialties that may also have a good sense of the pulse of what's going on. And this is a really strong motivator for staying connected with your interdisciplinary neurology care team at a specialized care center, because we try our best to be aware of what's going on in research, and places like where I practice, we tend to be involved in many of the research trials that are going on. So we're constantly reaching out to our own clinic population if they're eligible for trials to see whether they're interested. If you are not actively being followed at a care center, we don't know who you are. And so it needs to be your own impetus or one of your providers contacting us that lets us know that you might potentially be eligible for research. And so this would be a way to stay connected with your care team. If we can then go to the next slide. So clearly there are many challenges to being a research subject. And we know when we design these trials that we're asking a lot of people who participate in research. It's a tremendous time commitment, both for the subject themselves, but also for people involved in getting them to their research visits. And in pediatric research, that's a big burden on parents or other caregivers. This can lead to time away from school for the child participating, and work for the people that are also accompanying them to the visits. In the research trial designs, there's often some accommodation for this in terms of reimbursement for travel and for expenses related to getting to the visits that may be offered by the research trial. So this is an important question to ask as you're considering participating in research, if this is something that may potentially be a barrier for you. One of the things that is challenging about research design is that it's very strict about when things need to happen. And if we start to deviate from what we call the protocol, that leads to challenges with interpreting the results and the research not being as strong. So we really try our best to make everything happen according to a specific schedule. But that makes it very challenging because life comes up. You know, if you want to take a vacation for 2 weeks, is that during a time that you're supposed to have a research visit? And would it be permissible to do it a little earlier or a little later? These kind of issues are constantly coming up with our research families, and we're trying to find ways to come around if there's a snowstorm, anything that can come up to disrupt things. It's a tremendous amount of testing often, and in SMA trials, this tends to be things that people are already familiar with from clinic, like motor function measures and other things, but sometimes there can be things that wouldn't usually be part of your clinical care or would be more intensive than would be typical. We've had trials that have asked muscle biopsies of patients or imaging or other things. And so this is clearly a major commitment in terms of the person participating, that they allow all of these things to be done, which really aren't influencing their clinical care. If there's overlap with the research procedures, we often do try to economize and make sure that we're using the results from the research-related testing to inform our clinical management, and not asking people to duplicate tests if we don't have to do that. But ultimately, it is in the context of industry-sponsored research, it's up to the industry sponsor whether results from a particular test can be shared more broadly or not. And it tends to be pretty permissive, but sometimes we can get into issues of not being able to share these results with other providers involved in the care. One of the biggest risks of research, of course, is that we're still studying the medication and we don't know all of the potential safety risks associated with the particular intervention. And so this is a tremendous risk that you're facing as a research subject. We do try our best to set up trial designs to be very sensitive for anything that might be related to the intervention that we need to be watching for. And then we also have a mechanism for reporting that very quickly to all of the investigators involved to make sure that we're being safe and that things change if they need to as research is going on. And then, of course, there's all the social and cultural factors that come into any decision that you're making as a person as trying to decide about whether this is right for you. And so when we're screening for a trial as investigators, we try our best to understand the context of the person that's interested in the research and be very clear about what's being expected. The key points that need to be understood as a research subject is that you always have the right to withdraw from a trial once you start. That's not penalized in any way, and so we absolutely would want to know the reason why, but there is no obligation once you start. You can stop at any time. And also that it does not in any way influence the care that you get at baseline. And so we very much will continue to give you the best possible care for your SMA, whether you choose to participate in research or not. We can go to the next slide, please. The basic design of research trials is standardized. We tend to go through several phases of research trial to try to understand an intervention. The first phase is usually focused mostly on safety and tolerability. Sometimes this may even be in healthy subjects rather than in people affected with the disease. Then we go to phase 2, where we start to look for whether there's some sort of effect related to what we're doing, but still focus very much on safety as well. And then the gold standard would be to then do a phase 3 trial where we have a placebo arm and we have an intervention arm. Some people get the drug, some people do not, and we compare those two in the exact same way over a period of time. In the context of some of our newer approaches that are coming into the therapeutic arena, this isn't possible to do because they're much more complicated in terms of the way that the design is done. A good example of this would be gene therapy. There's been many guidances from the FDA about specific instances where this level of 3-phase design may not be the way that you need to do your trial design, and new sponsors are constantly interacting with the FDA about what level of evidence and how best to design their trial to meet the expectations that they may have. So it may be that even a single center design is sufficient in some instances, or multi-center designs but with no placebo arm, because it's too obvious who's gotten the intervention once you've given it that you can't actually have a placebo in some situations. And then once we're getting close to potentially being able to prescribe this for patients, some sponsors will choose to have an earlier expanded access program. We've had that happen in multiple SMA therapeutics recently. There's also a concept called compassionate use, which is a little different. But the key point here is that this is still done under a research regulated environment, and so either an individual provider has to decide, I think this patient needs access to this medication, and comes up with their own specific protocol of how they're going to do it and gets approval from the FDA to do that. Or there's a larger program that's designed with a similar protocol for multiple investigators and sites to be involved. But this has significant risk to it because it's not the same kind of formal trial design as what was happening in the earlier phases. And so if anything were to come up in one of these subjects, it still gets reported to the FDA, and it can actually affect the approval of the drug at the end of the day if there's a new safety concern or some sort of efficacy issue that wasn't previously identified. And because the investigators in this sort of design aren't always the ones that were embedded in the original research, there can be some issues related to site variability or investigator variability related to this. And so these kind of programs are available and they are regulated, but not always for every medication. Once we get it into the clinic, we then will do some post-marketing kind of research sometimes in conjunction with sponsors, where they try to do registry work or other kind of extended trial designs where they still try to understand how these interventions are affecting the people that were involved past the original duration of the designed research trial. And then we also have other research that is purely natural history, where we're not doing any active intervention. We're just trying to understand the disease and doing standard measures to be able to do that. And I know many people in the SMA community have been participating in these kind of trials for a long time. So the next slide, please. The bottom line here is that I think that people that participate in research are very noble in one sense, because they're taking a risk and putting themselves through a lot of stress and a lot of invasiveness that they wouldn't normally have to do. They do have the potential benefit of having early access to something that may be impacting their disease in a positive way, but I have tremendous respect for those who are subjects in trials that I participate in, and try my best to lessen the burden as much as possible. And I know that sponsors of trials have that same objective in mind, counterbalancing their agendas in terms of trying their best to get the science forward and getting things into the community. So that is what I wanted to lay forward, and now I'll transition it over to Dr. Castro to go over what's going on now.
Perfect. Thank you so much, John. So first, I have to say that it's not enough time to discuss all the studies, and it's a good problem to have. We have right now a lot of things going on. So what I'm going to try to do is go through the ones that are active or are more recent. And then at the end, when we have questions, I will be open to answer more specific questions about other trials. So next. These are my disclosures. Next. And I start with this every time I give a talk like this. I start thinking, I like to think about therapies in a way of what part of the problem where SMA, SMA affects not only the nerve, it affects the muscle, it affects the junction between the nerve and the muscle. So I like to think, what is that those therapies are targeting? So the first group of therapies, like you see in that yellow box, are the ones that we call SMN targeted therapies with SMN2 splicing. So meaning we're going to have somehow increased levels of that SMN2 protein that is left in the body. Within those, we have nusinersen, we have risdiplam, there is another one in Europe called branaplam. And now we'll try to discuss really quick about pretty much what is going on. Then subsequently, I will talk about gene replacement therapy that is also acting into the anterior horn cell. Then there are some muscle enhancing therapies, and I think we'll be pretty much done at that point. So let's go to the next one. So modulation of the low functioning SMN2 or backup copy. Like I said, there are two big ones right now, oral risdiplam and intrathecal nusinersen. Within risdiplam, there are different several studies according to the age of the patient or if the patient is pre-symptomatic or not. In general, Firefish is a study that is done for patients less than 6 months of age who had a confirmed diagnosis of SMA. This study is not enrolling, that's why I'm not going to spend time on this one. But again, I can answer later some questions about results and so on. We will go to the next one. Rainbowfish is the one that I wanted to talk, because this is a study that is currently recruiting. It's an open label study for patients that are asymptomatic. So kids that are born that we know have SMA somehow, or they got diagnosed with newborn screening, and they are between birth to 42 days of age. They have to be genetically diagnosed with SMA, and they can have any copy number of SMN2. So they can have from 2 all the way to 4, 5. All infants will receive risdiplam for 24 months, and they will follow for 3 years as an extension phase. The target enrollment is 25, and I know that this is a global study, so it has been already enrolling patients, but this is a good possibility for some of those new patients that are diagnosed. What is the exclusion criteria? If the patient has been using or has used any of the medications that can modify the SMN2, meaning if the patient has had received nusinersen or if the patient has had received gene transfer therapy, that will be exclusion criteria. If the patient requires invasive ventilation, tracheostomy, or awake non-invasive ventilation, meaning the form of BiPAP. or if the patient has other disease or other syndrome that is associated in that case with SMA. So those will be the cases where, unfortunately, they will not be included. Next. Then when we look at the intrathecal form, nusinersen, currently there is not a trial actively enrolling. We'll talk about the newest one that is going to come later on, DEVOTE, or it's already coming. So next. This is just, I like this slide because it shows all the studies that have been going on since back in 2011, really. We started first with what we call open label studies, meaning we will give medication to everybody. Then later on, we did what we call randomized studies. So some patients were given medication, some of them were not getting medication. And then finally, today, what we have left out of all of this study are really two studies. One called SHINE, and as you see, all of the ones that came from 2011 all the way to 2015, 2016 end up in SHINE. And then the other one that is still ongoing is called NURTURE. And NURTURE is the one in pink and in the lower part of the slide. And NURTURE is for the pre-symptomatic babies. So they are already, again, they are not enrolling for any of these studies, but it's important that we're following them over time, over many more years. Next. DEVOTE is the one that I get a lot of questions from parents, and it is open to enrollment. DEVOTE is going to have different parts, so different groups. Part A is going to be for patients that have had signs or symptoms of SMA older than 6 months of age, so meaning called later onset SMA. So these patients will have to be between 2 to 15 years of age. Part B will have two groups. One that is symptoms less than 6 months of age, and then a second group that is with an onset more than 6 months of age. Within this subgroup, they will have to be between 2 to 10 years of age. They should be able to sit independently, but they have never had the ability to walk independently. And there is a score that obviously when patients go through a screening process, there are scores that are done, or functional scores. As Dr. Brandsema was saying, we try to choose a population that we know it's going to respond to treatment. So these are the numbers that sometimes you're going to hear, because those are the scores we want to start with. Then there is a part C that is for adults. It's participants that are older than 18 years of age, but they should be able to walk. They could be currently on nusinersen treatment at the time of a screening, with the first dose being at least 1 year prior to the screening. So that will be the inclusion criteria for this study. Next. The exclusion criteria, these are for all of them, A, B, and C. And these are the important things that we have to look into. One of them, the first one, if the patient has any active infection that is requiring any therapy with antiviral medication or antibiotics at the time of screening. It doesn't mean that if you have one infection right now, it doesn't mean you cannot screen later on, let's say in 3 months. The next one is a shunt. If the patient has had a shunt placed in their brain, or if has had a catheter that sometimes has been used in some places, like a catheter called Ommaya reservoirs and so on, these kind of implanted devices are a cause of exclusion for this study. If the patient has been hospitalized for surgery or for lung disease, or has required nutritional support within 2 months of the screening period, or if there is already planned surgeries coming. So sometimes we have seen patients that unfortunately we wanted to get them in the trial, but the scoliosis got really bad, really worse, so we have to go ahead and move to do surgery, so that will exclude them for the trial, and this is the case. If they have had onasemnogene, meaning Zolgensma, within 6 months prior to the screening, that's another exclusion criteria. Next. And then there are some exclusion criteria that are for Part A and B, and one for Part C. So for Part A and B, again, if there is invasive or non-invasive ventilation for more than 6 hours during a 24-hour period at a screening, if there is medical necessity for a gastrostomy tube, a G tube, or if the patient has had received investigational drug for treatment of SMA within the 30 days prior to screening. And the Part C, if the patient is participating or an administration of nusinersen in another clinical study. So if the patient is already getting nusinersen, it may be exclusion for Part C only. Next. So we go back to the initial slide, and this is just to go back to where you look at the areas that are targeted by the medications. So now we're going to look at gene replacement therapy. Unfortunately, again, there is nothing actively enrolling, but there is one that I will show you next that is called STRONG. And the reason I put it here is because right now this trial is on hold, but we hope that it will be open again and we will be able to start enrolling patients. So STRONG is for patients with type 2 SMA. And the difference between for gene transfer therapy, given the commercial or the prior trials, is that it has been given through the vein. In this case, this specific trial doesn't give the medication through the vein, it gives through a lumbar puncture, through a spinal puncture, like nusinersen. And it's given only once as well. But right now, because there has been some findings in the animal model, not in people, but in the animals, they wanted to hold on. The FDA is looking into it, and hopefully it will open again. We know that there are 30 patients already enrolled, and we have some results from those patients. So there is a dose A, that is a low dose, and a dose B, there is a mild dose. And we know that at least for the dose A and dose B in the younger group between 6 to 24 months, 7 out of 6 patients have gained milestones. 1 of them stood independently and 1 is walking alone. These are patients with type 2, remember. So these are patients that obviously will not be able to walk otherwise. And then for the second group in dose B, between 24 to 60 months, we have 3 out of 12 patients have gained motor milestones and at least 1 walk with assistance. For dose C, there are only 2 patients enrolled currently. And as I say, because the trial is on hold, the enrollment is not complete yet. Next. Then muscle enhancing therapies. It's not enrolling. Currently, the enrollment was completed, but the reason I wanted to put it there is because it's another target area that it's explored currently. So the study is with a medication called SRK-015. And next. This is a phase 2 study with active treatment to evaluate the efficacy and the safety of this medication. The phase 1 was done already in patients, in healthy humans. So phase 2 is now with patients. The SRK, the medication, is something called promyostatin. Myostatin is a substance that we have in our body that controls the growth of the muscle. When you inhibit that, it allows the muscle to continue growing. So the hope is that we can target not only the anterior horn cell, the nerve, but also target the muscle to make it hopefully stronger. It is an IV infusion. It's every 4 weeks. And like I said, it's not enrolling right now. It was completed, the enrollment was completed really, I believe, early this year. And hopefully the completion of the study will be in April of 2021. Next. There are 3 cohorts in the SRK. The cohort 1, it's an open label study, and this is for type 3 patients. There is a cohort 2 that is also open label, meaning the patients are getting the medications we know they are getting. That is for type 2 and non-ambulatory type 3. And there is a cohort 3 that actually, those are the patients that I have in my side enrolled, where they are randomized. So some of them are getting 20 milligrams per kilo, and some of them are getting 2 milligrams per kilo. And this is for patients with type 2 that are less than 5 years of age. Next. We know that there is some preliminary data for what we call pharmacokinetics and pharmacodynamics. It's just related to how the medication is distributing in the body, how long is it staying in the body, and so on. There are 29 patients, 12 in cohort 1, 8 in cohort 2, and 9 patients in cohort 3. It says that it's a dose-dependent increase of the level of myostatin, meaning that the dose have been given between 2 to 20, and the dose will give according to the level of dose, it will give you a higher level of the myostatin in the blood. There is also a fall increase from baseline in the serum myostatin levels in the first 4 weeks. That is very similar to the phase 1 trial that was done in normal, healthy individuals. And what is very important is that there is no clinical significant safety signals that have been observed. So we have not had anything that makes us concerned that this medication is not safe. Next. And I think that's it. So again, we will go through some of the questions, and unfortunately, there is never enough time to talk about all of these trials, but please feel free to ask more specific questions if you have any.
Great, thank you. Thank you both so much for the incredibly informative presentation, Dr. Castro and Dr. Brandsema. Like you said, there's a lot of information that we went through, and we see a lot of questions coming in, so we want to get to as many as we can, so we'll jump right into the questions. Our first question we have for you both is someone able to participate in SMA research and or clinical trials if they are receiving any SMA targeted medications, and can this be done through insurance?
Right, so I think this is going back to the concept of inclusion and exclusion, that some trials permit you to be on other therapies and add something on in addition, and others want somebody to be, in a sense, pure and not exposed to something previously before they start the intervention in the trial. So it really would be specific to what you're asking about, but some people who are receiving nusinersen commercially, for example, could go into the myostatin inhibition trial that Dr. Castro was talking about, for example. So there's an understanding that some of these things could potentially be complementary, and we need to study whether together there's more benefit than either one by themselves, and also are there any safety issues that might come up in the combination that weren't there for either one independently. And combination study is a very important concept. There are some discussions happening about early discussion of combination trials related to some of the approaches, and we've already had one with the myostatin inhibition, but this is all stuff that's going to be on the horizon.
Yeah, because the patients that we have right now in the myostatin inhibitor trial are patients that are continue getting their nusinersen through commercial means. So yes, they can. But if you look at the specifics, Rainbow Fish, that is the one for pre-symptomatic babies, yes, they don't want the babies to be on any other medication. But if you look at Devote, that is the one that is going to come with a higher dose of nusinersen, it allows for the patients, some of the patients, to be on a medication. So another site that I recommend a lot, and I always give it to the families, is called clinicaltrials.gov. And I think it's an important site to remember and to look into it. You can just type the disease you're looking for, SMA in this case, and it will bring all the trials that are ongoing, including the natural history studies and so on.
Great, thank you both for addressing that. The next question we have is, do you see any impact that COVID-19 is having on the progress of existing SMA clinical trials? And has enrollment or patient visits slowed down and affected the endpoint measurements?
So I will start. Unfortunately, yes, we're talking about this yesterday. The treatment itself, in my institution, for example, we are treating all the patients that are receiving nusinersen. They keep getting their nusinersen every 4 months. So that has not been affected. The problem is that when you think about a research patient, you have to evaluate so many things, including physical therapy. You have to give them medication. You have to do labs. So sometimes those kind of visits may take 3 days. 2 or 3 days. So we're trying to minimize the exposure of those patients to the hospital. And right now what we're doing is we are limiting the visit pretty much for treatment. We're holding on the physical therapy evaluations. So we're getting the treatment and the labs pretty much. And the physical therapy, we're just gonna start going back to do it again. So it will affect the study in a sense, because you're gonna have those points that are gonna be missing in the data.
Yeah, here in the United States, there's been guidances from the National Institutes of Health, from the FDA, and from the CDC about how to best do this in this crisis that we've all been in globally, but it really comes down to an individual site decision and an individual investigator decision sometimes in terms of what we collect. And so for the protocols I've been involved with, it was extensive communication with the different sponsors of the trials or the NIH if it was an academic study, deciding which procedures were truly essential, whether it be delivering a medication or monitoring either for safety primarily or efficacy, and which things could be deferred for a period of time. The challenge being that we're all constantly adjusting in this new world that we're in and don't have a clear timeline of when we're going to be out of it. And so this is still in flux, but we're trying our best to keep protocols active, and we haven't abandoned research in this context. It's just needed to be modified with the best intent possible from everybody involved.
Wonderful, thank you. I know you touched on this a little bit about combination therapies, but can you share any more about current data regarding combination therapies that induce SMN transcription and others that focus on muscle function?
You can go ahead, John.
Well, yeah, I think we already do this sometimes in the clinic, you know, that we'll have people who are on nusinersen or have received gene replacement therapy, and then we put them on an oral form of albuterol, for example, to help their fatigue, you know, and so we're always trying to think of ways that we can optimize people's function. And the more therapies that we have approved for SMA, the more that we can start to think about how to use them together. The challenges being, of course, that we want to be sure that we're being safe in our choices, that we're not making more risk for the person by potentially having toxicity that we weren't anticipating. And then there's also just the pragmatic aspect of how do we get all these different therapies to people. And some of these treatments involve quite a bit in terms of getting the therapy into the person and also, frankly, the cost. And so to be able to come up with protocols that use combination is daunting in some senses because of that. And so we will, as a community, keep working on this, but it's sometimes not as straightforward as you might like it to be. We're always trying to strive to give the best possible outcome for somebody by putting them on the right combination of treatments to optimize their life living with SMA.
And we were hearing yesterday a couple of reports of combination therapy. So there are a very small number of patients that have been treated or that now are reported. There are probably many, but not reported yet that we know. And it's very hard to figure out which one is giving what. Was the patient already improving and then you give a second therapy? Is that second therapy really causing a rapid improvement, or it was the other one that was still helping? You know, so it's very hard for us to really say something about it until more studies are coming. Hopefully more studies will actually come, studies that are regulated, and they are going to look into functional testing and let us know if really there is a difference or not. I do agree with John. I mean, I try to do as much as I can, but the insurance limit many of our options. So what I have found is that the cases that I have with double therapies, because they were on a research trial before and now they came out of the trial, so now they were able to get a second one. And I was able to switch some more switching, not combination, but switching therapies in some of my commercial patients, because I found that as my decision, it was a personal decision, it's a decision with the family, it was that we were not seeing the response that we wanted to see, or the response that has been reported. So we will see how it goes, but hopefully, as I said, there are different targets, there are different areas that can be targeted, and hopefully we'll have more therapies as we go.
Great, thank you. Another question we had come in is, what is the reason for exclusion in the DEVOTE study if a person has Ommaya reservoir?
You know, I think that I will say, I'm not from Biogen, but I will say that the problem is that there are no studies showing how Ommaya reservoirs work. We don't really know. In my clinic, for example, we have 6 or 7 patients from my colleague that are getting it through the Ommaya reservoir, but we don't understand how the medication is distributed or how long does it stay in the reservoir. There are many things, many questions that are not answered, that I think they have to take that variable out of the study to be able to have a more clean study. That will be my opinion, John. I don't know if you think something different.
I would agree. It's about this concept of trying to keep the population studied as pure as possible, and we don't know the variability related to that administration method. I just also wanted to make it clear for those on the call, I'm not sure if you stated, Dr. Castro, that the DEVOTE study is studying a higher dose of nusinersen than is currently commercially available, and so that's the intent of the trial, is to give more of the medication at each of the doses and see if that's better tolerated based on some animal research that was done showing that it is likely well tolerated and may lead to a different pharmacokinetic profile. But we need to do the research to be sure about that.
Great. Thank you both. Another question we have is, do you think muscle enhancement treatment would work if too much nerve damage has been sustained, such as in older SMA patients?
That's a great question. It's a question that I get often, actually. You know, we don't really know yet, but if you think that a lot of muscle, it's not present anymore. Whenever SMA happens, obviously over time, there is destruction of that muscle and there is fat replacement. So if the muscle is not there, it's going to be hard to try to stimulate the muscle that is really not there. But there are many things that we still don't understand. You know, there are many things that that we may be able to target even before the muscle is present. So I don't know. I think that we don't have a clear answer for that. I don't know, John.
It's a really great question. I sometimes talk about problems in the lower motor neuron as being like an assembly line to patients, you know, that if you're missing one of the key pieces in the overall assembly that you're trying to make, what you're making isn't going to be as good. So yes, you can get some other people to do another piece of the work, but if you're missing that key piece, is that actually better? Making more of something that's still critically missing something that's important for its function. And so I think there probably is a role for muscle enhancement in somebody who has a lot of disconnection to their muscle through denervation, but there's probably a point at which that's going to be stressful and harmful too, and where is that sweet spot of who's going to benefit needs to be so carefully studied in different populations to make sure that we're doing the right thing for everybody by offering a particular choice. But we've been surprised before. There was a lot of skepticism about whether an intervention like nusinersen might do much once somebody's had a lot of denervation, and there's been some clear evidence now that people who were severely weak at baseline benefited from that treatment. So I think we do have to be brave and try and learn together about it and try.
Great. Thank you both again. So the next question we have is, when, if you have any idea, do we expect the hold to be lifted by the FDA for the DEVOTE trial so that further patients can enroll?
The DEVOTE trial is not on hold. The one that is on hold is a STRONG trial for the gene transfer therapy. So DEVOTE, the higher dose of nusinersen, it's actually enrolling already in some sites. Some other sites like mine, we're still waiting for all the process to finalize. So DEVOTE is ongoing. STRONG is the one that is actually on hold because the animal model has shown some changes that were worrisome, and they wanted to, the FDA and the company obviously wanted to make sure that they were okay to continue enrolling people.
Great. Thank you, Dr. Castro. I think we'll do two more questions. Is there any science around the peripheral need for survival motor neuron protein?
These are really great questions. So yes, on the basic science side, there's been quite a bit done in mouse models and other things, looking at other tissues besides the motor neuron and how they may be affected by having too little SMN, or survival motor neuron protein, which is the problem everybody living with SMA has. There's been also some look in people living with SMA at some tissues and how involved they are. And the evidence is kind of mixed, and there's not like a clear yes, absolutely, there's this problem that everybody with SMA has, but there's some trends, and especially in the more severely affected people, you can see some other tissues being involved much more frequently. And so this comes to a question of, if you're then giving back the SMN protein, are you giving that back to the entire body the same way, or is it dependent on how you're giving it, only getting mostly to the brain tissue or the spinal tissue, for example, and not so much to other tissues like liver and other things? There's also factors related to the tissues themselves in terms of how well they hold on to an intervention. Maybe they turn over and they won't keep whatever you give it. So there's a lot of variables at play, and we're trying to look into this on multiple levels on the research side, but there's still a lot to be learned.
Perfect. Thank you so much, Dr. Brandsema. So I think this will be our last question. Is there a list or database a patient can be added to sign up for trials to be considered for any of the trials, whether they live in the US or overseas, or should they just be checking with their provider and the clinicaltrials.gov website?
What I always recommend to the patients, one very important reason to come to clinic, it's besides the standard of care, it's to be able to be part of that clinic. Because if that clinic gets a research protocol, what we go is we screen our list, we go through the list, and we pick the patients that we think are going to be qualified, that they will qualify for the study. So I think that getting to your provider, making sure that if your institution, the institution you go is an institution that actually they perform research, but if it's not contacting all the institutions, is perfectly okay. So I don't think there is a specific list, a national list, but there is definitely, each of our institutions keep a database of patients.
But if you're in a situation where it's hard for you to get to an SMA care center of excellence, whether it be where you live or your insurance coverage or whatever else, maybe there's an opportunity to do less frequent assessments with that kind of care center, and then more frequent with whatever your usual care team would be, and sharing care that way. That's one option. Also, if you've never been able to get to an SMA care center, you can always reach out to a sponsor of a trial or one of the sites that you're interested, and that site will interact with you about whether or not you might be a good candidate. So that's where listings like the clinicaltrial.gov or going through Cure SMA or MDA listings is important, because you can reach out to individual sites, and we try our best to work together with the sponsors and the other sites to ensure that we're being fair and equitable about access, but also that everybody that's interested that may be able to participate is being engaged properly. It does help quite a bit to have your care established at a trial site, but it's not exclusive. We do bring people from elsewhere for our trials.
Great. Thank you both so much again for getting to and addressing many of those questions that have come in, and also for your time and presentation today, Dr. Castro and Dr. Brandsema. And thank you to everyone who participated and listened in on today's webinar. You will be receiving a follow-up email with a survey link, and we would greatly appreciate any feedback that you have. We are thankful for the support from our sponsors for making this virtual SMA conference possible. And if you have any additional questions after this webinar, please reach out to conference@curesma.org. Have a great day, everyone. And there is one more webinar happening this afternoon, and then tomorrow is our last full day of the virtual SMA conference. So please check out Cure SMA's conference webpage for all the events tomorrow. And thank you all again so much.
Thank you.